Proteins are made from mRNA, which are basically copies of DNA made specifically for the purpose of protein synthesis. This is a piece of DNA. It is composed of two strands, both of which are made up of the nucleotides adenine, guanine, cytosine, and thymine. On the DNA strand is a sequence called a TATA box, which is made up of TAT AAA nucleotides. This is where transcription begins. TF2D is made up of TBP, aka the TATA box binding protein. It binds to the TATA box and bends the DNA. TF2A stabilizes the binding of TBP to the TATA box. TF2B binds and determines the start site. TF2F has polymerase II bound to it, which binds to the start site. CTD is bound to the polymerase. TF2E binds and recruits TF2H. TF2H is a multi-complex protein consisting of a helicase and kinase. The helicase unwinds the DNA. The kinase phosphorylates CTD. The strand transcribed by the polymerase is the pre-mRNA. It is composed of introns, which are blue, and exons, which are black. A 7-methyl G cap is added to the 5' end of the RNA. This protects the 5' end and enhances export, translation, and splicing. The introns of the pre-mRNA need to be spliced out before the exons can be translated into protein. This is done by small nuclear ribonucleoprotein particles, also known as SNRNPs. The U1 SNRNP binds to the 5' splice site. The U2 binds to the branch site that requires ATP hydrolysis. U4, U5, and U6 join as a complex that binds through ATP hydrolysis. U5 binds the 5' splice site, then the 3' splice site. U6 catalyzes the splicing. U4 prevents U6 from catalyzing. When U6 unbinds U4, it base pairs with U2. U1 and U4 are released, and U6 is allowed to catalyze the splicing. Splicing occurs via two transesterification reactions. The 2-hydroxyl group of the adenine in the branch site makes a nucleophilic attack on the 5' phosphate of the 5' intron. This displaces the 3' end of the exon 1 and a lariat structure is formed. The U5 SNRNP aligns the released exon number 1 with exon number 2. This allows the free 3' hydroxyl group of the X1 that was released to attack the 5' phosphate of the second exon, forming a bond. In cotranscriptional splicing, this occurs before transcription is fully terminated. When the polymerase reaches the nucleosome, it cannot transcribe anymore. The sequence AAUAAA forms a stem loop, allowing the polymerase and the mRNA to be released. ATP is required for the addition of the poly A tail, which protects the 3' end of the RNA. It also enhances export, translation, and allows for alternate processing for different mRNAs. Alternative splicing is utilized to create a variety of genes. The strength of splice sites determines if an exon is included or excluded. By doing this, a variety of mature mRNA genes can be made from one pre-mRNA.